Hello, let us look at the microbial production of amino acids. Looking at the history of amino acid production, the first marketed amino acid is monosodium glutamate, and the production of monosodium glutamate or MSG started back in 1907 in Japan. So, what is MSG? MSG is marketed in the brand name Ajinomoto. So, this is a food additive. This particular amino acid is having demand even now. Let us look at the application of amino acid. It is used in food industry, it is used as a feed additive, and it is used in the pharmaceutical and chemical industry. In food industry, MSG or monosodium glutamate is the mostly produced and the mostly used one. So it is used as a flavor enhancer and it is used in different cuisines like Japanese, Chinese, Korean cuisine, mostly used in these cuisines. Then aspartame. Aspartame is a combination of aspartic acid and phenylalanine. This is an artificial sweetener. So this is being used by people who are health conscious, who want to reduce their calorie intake. Okay, are using aspartame as a sweetener, and the people who are suffering from diabetes, because this is a zero calorie sweetener. Then tryptophan plus histidine is used to prevent oxidation of milk, and cysteine is used to prevent the oxidation of fruit juices. Certain amino acids are added in food like bread so as to improve the nutritional quality of these food items. Amino acids are added in the animal feeds. Usually, limiting amino acids are added such that the animal is getting these amino acids from the animal feed rather than from natural sources. And in pharmaceutical, amino acids are used in infusion fluid, which are given to the patients after, like, post operation, or in some condition the patients are given infusion fluid, and this infusion fluid will be having amino acids in them. It is a component Amino acid is a component of many herbicide. One example of the herbicide is glyphosate, and amino acids are used in leather industry, cosmetic industry, etc. So how is amino acid production done? Amino acids are produced by three different processes. It can be extracted from protein, and the protein can protein can be extracted and hydrolyzed, and this protein hydrolysate can be used for amino acid production. We know that amino acids are building blocks of protein, so this protein hydrolysis can result in amino acids which can be used, or else artificial synthesis using enzymes, the chemical synthesis can be done, or microbial production can be done. So we are interested in the microbial production of amino acid. So how are different microorganisms used for the production of amino acid? Let us look at this. You can use microorganism for the direct fermentation method, where you're using the microorganism, you're growing the microorganism using certain raw materials and carbon sources like glucose, etc. And after the microorganism or during the growth of microorganism, certain amino acids are produced. These amino acids are extracted from the media, purified and used. Okay, or else the microorganism can be used to convert one metabolite into the required amino acid. So a one step or a few step reaction is catalyzed by the microorganism, not the complete fermentation is done. So only selected reaction for amino acid production is done. For example, glycine can be converted into serine using microorganism. Then microbial enzymes can be used rather than using the complete microorganism, or you can use immobilized cells and immobilized enzymes. Okay, for amino acid production, one example is amino acid dehydrogenase. This is a particular enzyme that is isolated from Bacillus megaterium, this bacteria. So this particular enzyme, what it can do is, this can be used for the production of L-amino acid from alpha keto acid. So alpha keto acid is converted into L-amino acid with the enzyme amino acid dehydrogenase. So this enzyme, this important enzyme, is immobilized, okay, in a matrix, and it is used. Some examples are alanine production from pyruvate using this particular immobilized enzyme, leucine from alpha keto isocarproic acid, phenylalanine from phenylpyruvate. Now, looking at the microorganism used and the strain improvement of these microorganisms. The very first microorganism uh, showing amino acid production, promising amino acid production is Cornybacterium. This was the first discovered microorganism, Cornybacterium glutamicum. And later on, different other microorganisms were also used, which was further used for the fermentation process by which amino acids are produced. But these wild organisms, whichever is isolated from nature, they produce the amino acid which are needed for them for their growth, their cell wall synthesis, etc. So the overproduction which we are looking for is very, very rare in the wild species. Only with if lots and lots of amino acids are produced, overproduction is there, we can harness these microorganisms and use them. So what is done? Whatever's interesting microorganism producing amino acids are selected. These organisms are improved for the increased production of amino acids. So the different technologies that we are using for strain improvement are mutant selection, rDNA technology, and recombination. So coming to the amino acid production, the very first amino acid that we are looking at is glutamic acid, and the pathway involved in the glutamic acid production is shown here. So this pathway, whichever we are seeing, is glycolysis plus TCA cycle. That is, glucose is converted into glucose 6 phosphate and finally to phosphoenol pyruvate. This is glycolysis, and finally this phosphoenol pyruvate is entering into the TCA cycle. Okay, and so the carbon source of glutamic acid production is glucose. And these two carboxylation reactions, that is phosphoenol pyruvate to oxaloacetate and pyruvate to oxaloacetate, is an important rate limiting steps in the glutamic acid production. That means if this particular reaction is happening more and more, that means more and more glutamate can be synthesized. If this reaction is limited or if it is inhibited, the glutamic acid production is stopped. Okay, so these two steps, the first step, that is pyruvate oxaloacetate conversion by pyruvate carboxylase, and phosphoenol pyruvate to oxaloacetate conversion by phosphoenol pyruvate decarboxylase, sorry, phosphoenol pyruvate carboxylase. Both these steps require biotin. Okay, and coming to the TCS cycle, looking the alpha ketoglutarate that is formed during the TCS cycle can be converted into the amino acid glutamate with the help of the enzyme glutamate dehydrogenase. Alpha ketoglutarate to glutamate by this particular enzyme. That means if more of this enzyme is present, more of the amino acid is present. But alpha ketoglutarate can also go on and convert to succinate by a decarboxylation. Okay, so this particular step has to be stopped. We just want the glutarate, alpha ketoglutarate, to be converted to glutamate. We don't want the rest of the TCA cycle happening. Okay, so these are the important steps. So when we are looking at glucose, one molecule of glucose can give rise to one molecule of L-glutamate, and the efficiency of the whole reaction is 70%. And how are we going to regulate the glutamic acid production? The glutamic acid production can be increased by increasing the production of oxaloacetate and acetyl-CoA, which is going into the TCA cycle. So if an organism is having either pyruvate carboxylate or phosphoenol carboxylate carboxylase, it can produce oxaloacetate. Okay, but this efficiency, if either of the enzyme, only one of the enzyme is present, the efficiency of oxaloacetate production will be very, very low. But certain organisms are there which have both these enzymes. So in such cases, more and more oxaloacetate is produced. Okay, and hence more and more of glutamate is produced. So bacteria are selected which have two carboxylase enzyme which produce a significant amount of glutamate. Then the enzyme glutamate dehydrogenase should be very high, and the enzyme alpha ketoglutarate dehydrogenase should be very low, so as to promote glutamate production. So which are the microorganisms that are helping in glutamate production? These are the four important microorganisms: Cornybacterium glutamicum, Microbacterium, Brevibacterium, and Arthrobacter. Of this, Cornybacterium glutamicum is the mostly used one. And all these microorganisms have certain common properties. All these are non-motile gram-positive organisms. They require biotin to grow, so they have very low amount of alpha ketoglutarate dehydrogenase, or else they don't have this enzyme at all. And at the same time, they have a high amount of glutamate dehydrogenase, which is helping in higher production of the amino acid. So how is the strain improvement done? Okay, these are the microorganisms. So if you want to produce more improved, if you want the improved strain of the microorganism, how is the strain improvement done? C glutamicum can be improved such that more productivity is there. So the strain improvement is done by mutant selection of variety which can tolerate higher concentration of biotin. Higher those variety which can tolerate higher concentration of biotin are seen to produce more and more of the amino acid. And lysozyme sensitive mutants are also shown to have a higher yield. So these different types of mutant selection is done. So the carbon source can be refined sugar like sucrose, fructose, or unrefined sources like sugar or beet molasses. You can also depend on acetate, ethanol, methanol, etc. The nitrogen source, the most preferred one is ammonia. Okay, and the mechanism which have urease enzyme can use urea. Why? Because urea can be broken down into ammonia and used. But with ammonia, there is a problem. If higher concentration of ammonia is present or ammonia salt is present in the medium, it isn't going to inhibit the growth of the microorganism. So during the reaction, what happens? Very, very low concentration of ammonia is added into the fermentation medium, and as the fermentation is being continued, little by little of ammonium salt is supplied. The growth factor or growth factor is added as additive, that is biotin can be added at the concentration of 5 microgram per liter, and since this is an aerobic reaction, oxygen have to be supplied. If too little oxygen is there, if the concentration of oxygen is too less, it might lead to the production of lactic acid and succinic acid, and if higher concentration is there, it might inhibit the growth of microorganism. So how is glutamic acid production and recovery done? Glutamic acid, after it is produced, it is present inside the cell. So it is an intracellular amino acid. Okay, and hence there should be some method to release the amino acid into the medium. So for the easy release into the media, the cell membrane of the microorganism should be made permeable. The permeability has to be increased. So these are different methods by which the permeability can be increased. Like if there is a biotin deficiency in the medium, the cell membrane becomes more permeable. The cell membrane is less strong, so it became more permeable. So treatment with fatty acid derivatives can also increase the permeability. Oleic acid deficiency in the mutant, addition of penicillin in the medium, etc., can increase the permeability. The recovery. As the fermentation is completed, the cells are separated and the culture broth is passed through an ion exchanger. Okay, after it is passed through the ion exchanger, the glutamic acid is bound to the resin, and now this bound resin is eluted out with sodium hydroxide, and ammonia is released. With, with this sodium hydroxide, the glutamic acid will form the monosodium glutamic, which is then purified by passing through an anion exchanger again. And this MSG can be subjected to evaporation and crystallization. So these are the steps of glutamic acid production and the downstream processes. Finally, you're getting it as a monosodium glutamate, which is used.